Thank you, Morgan and Michelle, for having me. So yeah, it's my pleasure to be here this morning, happy Monday and start of the week. So I'm going to be talking about structured data and our use for it, in particular in rare genetic conditions. If I wanted to make this go, where do I point it to? Does it work?
I think maybe use the
I can use the mouse. Click. Great. Okay. So I'm here on behalf of a company, so there is a conflict of interest slide. So a lot of the things that I'll be talking about involve a commercial open source tool called FINA that I built a lot of my research on top of for my PhD and postdoc. It is a commercial tool owned by Gene42, which is the company that I'm now representing, but I've tried to focus much more on the research side of things here. Oh yes, it works now. Okay, so my background, just to give you a little bit of context, the lens that I see the world through, I'm much less familiar with the cancer world. So full disclosure of that up front. I'm coming from the rare disease space. There are a lot of similarities in different places, but also a lot of really big differences. So one of the things I'm excited about is coming here and getting to talk about some of the work that we've been doing, and then hear about any overlaps or potential collaborations, ways that this might connect into what's going on in the OICR community. So my background is that I started originally in the research world on the ENCODE project, which was a big project to map regulatory elements around the human genome. There I was working on downstream machine learning approaches, actually with a PI who's now here, Michael Hoffman, when we were back at the University of Washington in Seattle. And then I started working on genome analysis and variant prioritization. That led me to my PhD work, which was building on top of phenotypes, building matching algorithms and algorithms for finding similar patients with rare diseases. So these really rare, undiagnosed patients, trying to figure out what are distance metrics, how do we find other patients who might be similar, who might have the same disease in that same database, so that we can put those clinicians in touch with each other. That actually expanded out into a bigger project called the Matchmaker Exchange, where we were trying to do this across databases in a federated fashion. And then that became one of the driver projects for the Global Alliance for Genomics and Health. More recently, I was trying to, we rebooted a project led by in the patient-facing support side for people with rare diseases or families affected by rare conditions called Rare Connect, where we were trying to take those same approaches and algorithms and apply them to connecting families together. And more now I'm over at the commercial side. So that's just a very brief history of what I've been into on the research side. The main thing that we've been focused on for a lot of this work has been trying to increase the structure of clinical data that's captured, especially when we're seeing patients with rare or undiagnosed genetic conditions. So trying to move this migration from oftentimes faster paper records, which a lot of hospitals still use, towards digitized and then increasingly codified representations of the patient's symptoms, so that these things can be computable and interpretable across languages by computers in sort of semantic, structured ways. Click, click, click. Yes, okay. So this structured phenotypic information is really valuable for a number of different reasons. It makes a lot of really hard problems a bit simpler. We can use this for diagnosing patients or suggesting diseases based on known disease condition associations. We can share patient data globally. Once you translate a terminology into a different language, you basically unlock all data that's coded with that terminology to clinicians and researchers in that other language. There is the ability to, yes, okay, it's jumping a little bit. There's the ability to analyze exome and genome information by prioritizing those genes or variants based on the patient's condition when we have this phenotypic information, the patient's symptoms recorded in a structured way. And it can enable us to do patient or cohort discovery. And I'll get into a little bit of that as we go through. So one of the big challenges that we've been focused on was how do we actually build this sort of data into clinical workflows. So there are sort of two big branches of research work over the last decade or so in this space. And I would say one is taking the data that's already there, that's in EHRs, getting lots and lots of data of this form and trying to mine information from those records so that then you can compute on it. The approach that we took is a very different approach and complementary in a lot of ways, which is trying to build tools and interfaces that make it easy for doctors to collect that data up front as part of clinical care. There are a few big advantages to that approach. One, it can be a little bit easier or cheaper. You don't have to, obtain access to hundreds of millions of patient records. And two, you're actually more guaranteed that the data that you're getting is high quality, has been reviewed by a clinician, right? There are a lot of challenges with mining data where that data source may not be accurate. There may be typos or ambiguities there that aren't able to be represented when you're parsing that data out in an automated fashion. And so we have taken an approach where the specialist is still in the loop there. One of the main tools that we've used for this is a tool that was developed about 10 years ago. It's a vocabulary, or an ontology, as we would call it, for describing the physical characteristics of patients, primarily those with sort of dysmorphic features, things that are very often associated with genetic conditions. This human phenotype ontology was developed by a clinician turned computer scientist and nicely captures those sort of two competing goals when you're developing a terminology. It has to be valuable and useful from a clinical perspective, and it has to be valuable and useful from a computational perspective. And a lot of terminologies and ontologies are usually one or the other and rarely both. This has become the gold standard for describing patients with rare genetic diseases on the research side and is increasingly being used on the clinical side. So there are about 11,000 or 12,000 different terms, different specific features that a patient could have, and they're organized hierarchically. So it's actually an ontology in the sense that there are these semantic relationships of meaning between these different terms so that the computer understands, okay, if a particular patient has a coloboma, that is a type of abnormal eye morphology, which is a particular form of eye disease, which is an abnormality overall. So there's this top root that is the patient has something wrong with them, and it breaks down into more and more detailed terms as you go down. And these are then associated through large curatorial efforts with all known rare and common diseases curated from the literature and things like that. So there's a huge and growing set of data that we can use to compute on top of information when those cases are recorded in this fashion. So the tool that was developed in the research lab at the University of Toronto, so what is called FINA-TIPS, it started as a joint research project between the computer science department at U of T and the genetics department at the Hospital for Sick Children. So here we were trying to figure out how to structure this phenotypic information to improve the diagnoses of patients with rare diseases. Really, we started focusing on structuring data in 3 different categories. First, we structured phenotypic information using these HPO terms. Second, we're trying to structure family history information, building a pedigree drawing tool into it so that clinicians and researchers can record the patient's family history, who their parents are, who their siblings are, where is the pattern of features or diagnoses or genetics across this family. And then more recently getting into genomic information and structuring variant information so that we can actually then interpret and analyze it in the context of the structured phenotype and structured family history. There are a lot of workflow improvements that we try to do. Again, we're trying to do this somewhat difficult problem of building a tool that clinicians can use in practice and incentivizes them to use it. So one of the ways is by having growth charting, which is really common feature in electronic medical record systems, but taking those measurements that are really, really commonly recorded in clinical genetics, such as height, weight, head circumference, things like that, and making sure that any phenotypic features that are codified that are computable from abnormal measurements are automatically added. So again, these measurements can be pre-populated from connecting to the electronic medical record system. So streamlining this flow of information into codified structured phenotypes so that then we can compute on it. Similarly, this is text mining or concept recognition. So if you have textual data like referral note to genetics, you can suggest terms from that. We still take the approach of suggesting them. We still believe in the clinician being in that loop and generating labeled data for us. And that's sort of where we are in terms of the state of the field for this use clinically. But again, the goal is always to streamline the workflow of getting this data into a structured computable format. Increasingly, this involves collecting information pre-visit. So before the genetic counselors or the geneticists see the patient the first time, they can send out questionnaires asking the patient what their family history is and any information about previous testing. And we can use that to pre-draw the pedigree so that a lot of the time that would normally be spent in clinic going through and collecting this information is instead just spent reviewing it, sort of like the inverted classroom for the clinic. On the cancer side, we have a number of features built into the pedigree tool to try to help record different cancers over time, different cancers for all the different family members and the individual. And then because this data is now computable rather than being drawn on paper, we can convert it into formats that can be, for instance, connected into Bodicea or other cancer risk tools so that those risk curves or risk assessments can be computed on the fly dynamically. So again, once we have this structured data, we can use it to provide, for instance, real-time diagnosis suggestions based on known disease phenotype associations. We can look at all of those known diseases and say, here are those diseases that are the most similar to your particular patient, or here are those genes that are known to cause diseases that are similar to your particular patient. So we can suggest those things in real time and do quite well at that. More recently, we're actually doing that where you have this sort of virtual gene panel. So you have the patient's phenotypic terms that you've described for the patient already. You can generate a list of genes that are associated with some subset of those terms, apply a threshold, and then filter the patient's exome or genome results based on those gene lists that you've generated in the context of that patient record. So in terms of use right now, we have about 200,000 patient records that have been recorded in phenotypes, which with over 4,000 specialists and 100 or more instances. Phenotypes started as a research project. It received a lot of government funding to develop it. And so we've kept the core of the tool open source. So it's available for researchers around the world to download, set up, use it to manage their research study. Clinicians actually use it at hospitals. The company just comes in when it needs to be integrated into an electronic medical record system or something like that. They need a different license. But that means that people around the world, and in fact, most of the instances that we know of are people who download it and set it up and run it and use it totally on their own. People modify the source code and things like that. So it's all open source and available for that core. In terms of the overall phenotypes ecosystem, you can tell that there's a punchline there at the end. Phenotypes is used sort of across a spectrum of different places in healthcare and research for rare diseases. So it's used at hospitals where we do a lot of work integrating it into Epic and Cerner and other EMR systems. Those often feed into research clinics where it might also be separately used. So sometimes hospitals will have one instance clinically and one instance for research, and data gets explicitly pushed from the clinical side to the research space. So there's a barrier there where they're clear that no PHI is leaving from that clinical side into the research space. And then both of those feed into a lot of national and international initiatives for rare diseases such as Care for Rare, here in Canada, the Undiagnosed Disease Network in the US, SolveRD and RD Connect in Europe, and the UDNI for the undiagnosed disease programs across a bunch of different companies, across a bunch of different countries, use a platform that's built on top of phenotypes. So that feeds into this matchmaking unsolved cases aspect. There's a platform called Genome Central. That was a lot of the PhD work that I did, which is built on top of the open source side of phenotypes, which is one of the original nodes for the founders of the Matchmaker Exchange. So here we're really focused on patients that start out clinically, make their way into research and remain undiagnosed, make their way into these matching systems where clinicians can find other clinicians with similar patients. Really part of this loop, you have patients that you might be easy to diagnose, as they become increasingly difficult to diagnose, you get into gene discovery over on the right-hand side. So the Matchmaker Exchange is one of the driver projects of the Global Alliance for Genomics and Health. It actually started before the GA4GH started, and then we got sucked in as one of the core projects that was driving things forward there. So I was the technical lead for the Matchmaker Exchange for a number of years and sort of the liaison between that and the Global Alliance. And there what we built were APIs to connect a number of different nodes around the world, Genome Central here in Canada, MyGene2 and Matchbox and Gene Matcher in different places in the US, Monarch as well, Patient Archive in Australia, Decipher in the UK. These different databases that had all started springing up or had been around for a long time that were collecting case level information about patients with rare or undiagnosed conditions, all sort of around the same time started realizing that there was a big need for helping these clinicians find second families where they might have one case, but it's a very long process to go through and publish a case report and have some other clinician try to read that case report. Or what was increasingly being done is that people were just aware of these other databases, and they would email the curators asking, do you have a patient with this sort of, a gene, this sort of gene, or one of these 5 genes and this sort of phenotype? And instead, we built a federated network of these different databases so that you can do that computationally. You can enter a case into any of those different nodes and find matches across all of them. Genome Central is the one that we did here. And one of the things that we did was actually build into phenotypes the ability to push those cases, de-identified, to Genome Central so they can participate in this matching. So sort of built into the software is the ability to then funnel those undiagnosed cases in a safe and secure way off into this matching system so that they can end up being diagnosed. Even if you're not then actively engaged, your case might be that second family to some other person who has a case at some point. The last thing that I wanted to talk a little bit about is a new project that we're working on here in Canada, which is sort of a smaller version of this whole different ecosystem. It's part of a Genome Canada funded project, LSARP, a large-scale applied research project called Care for Rare Solve, which is being led out of the Children's Hospital of Eastern Ontario with Kim Boycott. We're building the genomic and phenotypic data repository for a number of cases. There are about 5,000 families, 4,000, I think, retrospective. So taking all of those existing different rare disease projects and pilots that have gone on around Canada and harmonizing all of that data and centralizing it, and then sequencing and analyzing 1,000 families prospectively, where they're in addition to exomes doing genomes for cases where those exomes were not diagnostic. They're doing methyl-seq and other omics data analyses for these patients, and then we're putting them into a centralized repository, which is built on top of phenotypes, so that that can feed into this workflow for Canada. So for sites that have phenotypes or other tools clinically, they feed into this research platform that's now centralized across Canada for rare disease patients. There's actually a thing up at the top left where it's connecting to another project that I worked on called I mentioned that at the beginning, Rare Connect. So collecting patient-entered phenotypic information so that we can compare that with clinician-reported information. And that's all feeding into this research portal, which has different levels of access control. So open aggregate access, controlled access for people who are outside of the consortium and need access, and then restricted access for people who are part of this consortium. And then for those cases that still remain undiagnosed, they again feed into the Matchmaker Exchange. So that is the summary. So phenotypes.org is the place for the open source version of phenotypes. There's a demo, you can download it and look at the source code there. And in terms of Global Alliance for Genomics and Health, the aspects of it that I'm the most involved in right now. So I worked in the discovery workstream, what's now the discovery workstream, for a long time in terms of the Matchmaker Exchange, but increasingly have been focused in the clinical and phenotypic data capture workstream, where there are new standards that are developing. In particular, there's a FINA packet standard for basically a bundle to send around this sort of information, right? All of these different arrows, we would like there to be some standard packet of de-identified, for research purposes, information about a particular patient that can be pulled from public, from mining of case reports, that can be entered by patients themselves, or that can be converted from clinical or research cases, and then trying to work on a new project to standardize some of this family history information. So pedigree drawing and family history is a big part of what our software does, and it turns out that there aren't a lot of very fleshed out common data models for sending this sort of information around that really capture all of the details that you would want from a genomic perspective. So we're trying to do a new project there where we, collect a lot of requirements, assess what's out there, and try to develop or put our stamp on some standard in that space that then ideally can connect into existing APIs like FHIR and others that are used for clinical and research data. That's it. So thank you. There are a lot of people who did a lot of the work that I got to talk about in different aspects of the research and company side. Most of this work, again, is the work that was being done is done in the research space. So phenotypes and Genome Central, a lot of people from Care for Rare, the NIH Undiagnosed Disease Program and Undiagnosed Disease Network were really early adopters and partners in this process. The HPO and Monarch team who curate these ontologies and a lot of the mappings and manage a lot of that so we can build these tools on top of it. And then the Matchmaker Exchange and Global Alliance for Genomics and Health, which you all know probably quite well. So thank you.